Hello, I'm Maryam Zehra. I'm a senior at the Harker School, and today I will be presenting my project investigating the effect of methylcobalamin on prenatal development using C. elegans as a model. I worked on this wet lab research at my high school under mentorship of Polygence mentor Samira Simkar. For decades, there has been speculation on the root cause of ASD in children and the recent global rise in diagnoses. Maternal intake, including vitamins, has been implicated. The goal of this research is to study the potential impact of the prevalently used vitamin B12 or methylcobalamin dosage, specifically on the CHD8 gene, commonly associated with ASD in humans. In the future, other researchers can build upon this research using organisms more akin to humans than C. elegans as a model. This study predicts that both a deficiency and excess in vitamin B12 intake will lead to the mutation of the CHD8 gene and will thus contribute to the development of the worm analog of ASD. The methodology involved dosing 3 groups of C. elegans with varying vitamin B12 concentrations, conducting thrashing analysis to observe motor activity, and performing DNA analysis to identify mutations in the CHD8 gene. The research contributes to understanding the genetic and environmental factors influencing the current rise in ASD diagnoses. ASD, or autism spectrum disorder, is a neurodevelopmental disorder that causes differences in the brain, social interactions, communication, and motor activity. Some developmental deficits include delayed speech and/or hyperactivity. While the immediate cause remains unknown, there is a correlation between an infant's diagnosis of ASD and maternal vitamin intake during gestation. Methylcobalamin, or vitamin B12 as it is commonly referred to, is often given to expecting mothers because this vitamin aids in the development of genetic material of the fetus's cells, the nervous system development, and red blood cell formation. While a deficiency of this vitamin could cause birth defects, an excessive intake has also been observed to lead to neural differences correlated with autism, according to an NIH study. The gene that is commonly mutated is CHD8, which provides the instructions for creating proteins that regulate gene expression by altering the structure of the chromatin packaging. C. elegans are unsegmented, soil-dwelling nematodes or roundworms. Typically hermaphrodites, these microworms are able to consume food and possess a nervous system. However, they do not contain a circulatory or skeletal system. They are commonly used in genetic and neuroscience research because of their short lifespan and large quantity of synapses and neurons despite their simple anatomy. Scientists are able to observe various neurological disorders using C. elegans as a model for the genetic mutations linked to autism because of the large presence of mutant DNA strands and genetic similarities to mammals. CHD8 is a gene which encodes gene-regulating proteins and chromatin remodeling. It is involved in the process of neural development, and when mutated, it has been commonly associated with ASD. If the vitamin B12 intake caused development of the worm analog of ASD, then the nematode's DNA would have the same mutation rate as the CHD8 protein, observable through RFLP analysis of the DNA. So for decades, there has been speculation on the root cause of autism. However, without genetic history, a fetus would develop ASD because of mother's intake. Often, alcohol was observed as being a common factor. However, there are also other substances, such as the necessary vitamins that a mother takes to ensure that the fetus fully develops. I chose to conduct research on the vitamin B12 because it is one of the most commonly taken vitamins and has been observed in a few past studies to be linked to neural differences. However, there is not yet enough evidence to distinguish between causation and correlation, so I decided to study this further. I hypothesized that a deficiency or an excess of vitamin cobalamin in the vitamin intake would lead to the mutation of the CHD8 gene, which, of course, like I mentioned earlier, is linked to ASD in prenatal organisms. Methods. So C. elegans. The C. elegans thrive in moist environments, including water and soil. So I created NGM/E. coli plates and kept the colonies in there at room temperature. Then I placed 10 adult C. elegans with a worm pick in each petri dish and waited for them to hatch eggs. For the worm pick, I used ethanol and flame to sterilize the tip. With a pipette, I removed the adult C. elegans and left 10 eggs in each petri dish. I prepared the vitamin B12 solution by mixing the powder with distilled water. Then I added 0 mg, 5 mg, and 10 mg of the vitamin B12 to each petri dish respectfully. So it was like a control, experimental, and experimental. And these eggs took 6 to 8 hours to hatch. Then I let C. elegans grow for 3 days and monitored them under a wide field microscope. I performed a thrashing analysis by taking the C. elegans and observing their motor activity under the microscope. Using a worm pick, I then transferred the C. elegans in the distilled water, timed them to see how many times they thrashed in 1 minute, meaning they moved their body from side to side. If one group thrashes significantly more than the others, they are more likely to have developed ASD. Next, I crushed the C. elegans exhibiting signs of ASD and amplified their DNA through polymerase chain reaction. Then I conducted RFLP gel electrophoresis of that DNA along with a strain of the CHD8 gene to see if the C. elegans have the same mutation. DNA fragments are divided during the gel electrophoresis based on their size. An electric current is used to draw DNA samples across the gel after they've been inserted into the wells or indentations at one end of the gel. Due to their negative charge, DNA fragments migrate towards the positive electrode. If the DNA fragments show the same distribution, meaning that their mutation rates are the same, then the CHD8 gene is present. If the fragments do not present with the same distribution, the C. elegans have not developed the mutated gene. CHD8 gene is commonly associated with autism in humans, fish, and worms such as C. elegans. This indicates that the methylcobalamin exposure to the prenatal C. elegans caused autism. Unfortunately, however, when I conducted this gel electrophoresis, the bands did not show up brightly enough because the fast blast dye didn't stain them properly. So I'm planning to do a repeat of this entire procedure with new colonies this month, and I will hopefully, using a different dye that is stronger, I will be able to analyze the DNA mutations effectively. So this data set contains 3 dosage sets, 0 mg, 5 mg, and 10 mg, each with a set of sample size of 10 C. elegans. The average increases and then decreases as the dosage increases. 85.5 mg for 0 mg, 116.8 mg for 5 mg, and 95.3 mg for 10 mg. Since the median values of 86 mg, 117 mg, and 94 mg respectively are close to the mean, it can be concluded that there are relatively symmetric distributions. As expected, both the standard deviation and the range increase with dosage. This indicates a greater variability in responses at higher dosages. There is a tail to the right because of the positive skewness for 5 mg and 10 mg, potential concentration of higher responses. The distribution has a lighter tail because of digigative cortosis values of all the dosages. Overall, the data suggests that a trend of increasing response with dosage. The increase in mean values and the positive skewness for the higher dosages indicate a rightward shift in the distribution. However, further analysis, such as statistical tests or a larger sample size, may be necessary to confirm the significance of these observations and establish a more robust understanding of the relationship between the dosage and the response. Additionally, the C. elegans were already swimming in liquid for a week and adapted to the buoyancy of water. So to make this data more reliable, the experiment should be repeated with the vitamin melted in the agar, therefore minimizing the liquid so that when the C. elegans are dropped in the new water, they can exhibit new movement. So in conclusion, this study investigated the potential link between the prenatal vitamin intake and ASD using C. elegans as a model organism. These nematodes were introduced to varying levels of vitamin B12 and observed through thrashing analysis for their motor activity. The results of the thrashing analysis revealed a trend of increasing motor activity as the vitamin B12 dosage increased. This indicates a potential correlation between vitamin B12 exposure and ASD-like traits in C. elegans. A subsequent DNA analysis aimed at identifying mutations in the CHD8 gene will be performed later for additional insights whether the microworms exhibiting increased motor activity have a higher likelihood of harboring the mutated gene. While the results suggest a correlation between the vitamin B12 exposure and ASD traits in C. elegans, it is essential to acknowledge the need for further research to establish causation definitively. The limitations of this study, for example, the small sample size and the potential influence of other environmental factors, must be accounted for in future studies. To enhance the reliability of the results, the experimental design should be refined by melting the vitamin B12 in agar initially to minimize the liquid exposure before the thrashing analysis. Additionally, this experiment should be repeated with larger organisms, such as zebrafish or rats. Overall, this research provided a baseline study regarding the complex interplay between prenatal vitamin exposure and neurodevelopment. It contributes to the understanding of the genetic and environmental factors influencing ASD epidemic and emphasizes the importance of further exploration into the potential factors contributing to the rising prevalence of ASD. Thank you for listening. I would just like to acknowledge Ms. Samira Sankara once again for her lovely mentorship and the people at my school who helped with my laboratory techniques. Thank you for listening.